row on the left-hand side. You can stand up. From right to left, we have Rachel, Willis, Tiziana, Tatiana, and Claire. And these guys were amazing to work with, just every single day, bring smiles to our faces. I'd also like to acknowledge Elena Lindy, who was just integral in keeping us coordinated and focused and on task in a very friendly manner. So before I get into the bulk of the presentation, let me take you into some background about what wetlands are, especially in the West Eugene area. So unfortunately, greater than 98% of the wetlands in the Willamette Valley have been converted to agriculture or industrial development uses and filled in. And as we continue to grow, and as our population expands in the Willamette Valley, so does our need for the crucial ecosystem services provided by wetlands for both our social and natural communities. They provide ecosystem services such as water filtration and purification, flood control, and they also reduce sediment flow into our rivers during high rainfall times. Moreover, wetlands are also an irreplaceable habitat for numerous rare and endangered species, some of which we ran into during our project, especially the Bradshaw's lomatium and Willamette daisy, which are endangered to the Willamette Valley and considered high priority restoration efforts. We also ran into a ton of camas and some tufted hairgrass, which is a very competitively dominant species in the Willamette Valley wetlands. So we are not the first people to study wetlands for the purpose of restoration. For 20 years, the Willamette Valley has been on the cutting edge of restoration ecology and restoration research. Our team's specific objective was to continue looking into the relationship between native species diversity and microtopography in these complex ecosystems. We carried out our study across 6 sites in the West Eugene area, 2 of which are considered restored wetland prairies, which is Dragonfly Bend and Coyote Prairie in blue. We also went to a number of remnant wetlands. In purple is a low-quality remnant wet prairie, red is moderate quality, and the 2 green sites, Willow Creek and Oxbow West, are considered high-quality remnant wetland prairies. So before I go any further, again, I'd like to stop and define what microtopography is for those of you who haven't spent 22 weeks figuring it out. So microtopography can be defined by very small scale variations in the height and the roughness of the terrain. And it can be formed naturally when surface water runoff, as well as excess flood water, is spilled over into these wetlands and forced around natural features like this tufted hairgrass here that I showed you earlier. As it gets diverted around this tufted hairgrass, over the years it'll cut rivets and channels into the ground, creating low points and high points in the ground. Now in restoration, microtopography can be emulated by disking and excavation. Disking is where you run blades through the ground and chop it up and make it kind of rough and bumpy. And that simulates what happens naturally over a period of several years. And what this does is create several unique niches for different plants to inhabit, which Audie's going to go into when we get into the technical stuff. So as a team, we hypothesized that the variation of microtopography is influential in determining plant community compositions, which is displayed by this flow chart over here to my left. And we can measure native species diversity across these sites based on 3 different calculations. And there are species richness calculations, species evenness, as well as the Shannon's diversity index calculation. So in theory, when native plants fill the niches provided them by the microtopography, they'll also help reduce the amount of introduced species that will colonize and establish themselves in these wetland areas, which is kind of what we're trying to do in a successful restoration effort. So I'm going to turn it over to Audie now, who's going to talk about the more technical aspects as well as the methods of our data gathering and analysis.
All right, hi guys. So over the course of 6 weeks, we went to each of the 6 sites and we had 3 different methods for collecting data. The first one you see for topography and water depth, we used an auto level, and along transects at each of our sites, we measured for topography, so elevation at these points, as well as recording the water depth. For plant community composition, we used a simple 1 by 1 meter quadrat made of PVC pipe. And we did this to look at the cover classes of different functional groups, which included grasses, forbs, bare ground, trees, shrubs, moss, lichen, litter, and litter is just dead vegetation that's hanging out on the ground. using the Dobermeyer cover class method. And then for vegetation height and litter, along the same points that we measured for topography, we used a point intercept method with the meter stick recording the highest point that the vegetation came into contact with along the meter stick, as well as measuring for litter and water depth height. So, after we went out and collected data, we went into our lab and slaving away over weeks of trying to find some significance in the data we collected, as well as data given to us by the Institute for Applied Ecology. This first figure that you see is a basic count, so species richness of the species that are found at each of our sites. And so the blue bars you see are the total number of species found. The red are the native species, and the green are the introduced species. And so our sites range, with Willow Creek having the highest number of species at around 40, and Oxbow West with the lowest amount of species at around 20. And what's important to note about this graph is the amount of introduced species, because it'll pertain to some of the explanations we have later in our presentation. So again, Willow Creek did have the highest at around 20, and then Coyote Prairie had around 4, so pretty good. This next figure you see is the Pelu's evenness indices that were calculated for each site. And basically what evenness looks at is how close in numbers each of the species are at each site. And after doing data analysis for this, our group didn't come up with any statistically significant results, and we didn't find any positive correlation within our topography data. Now this figure shows the Shannon's diversity indices that were calculated using the equation at the top of the figure. And Shannon's diversity indices is comprised of 2 components. So it takes a look at both the species richness and the species evenness at each site. So this ranges, well, first of all, for our study, 1.7 diversity index indicates that the sites are high quality, or not high quality, I'm sorry, highly diverse. And as you can see, all 6 of our sites have an indice over 1.7, with Dragonfly Bend at the highest of around 3.17, and the lowest, Willow Creek, at about 1.68, so 1.7. But what's interesting about this part of our data analysis is that the 2 green bars that you see, Oxbow West and Willow Creek, are considered high remnant or high quality remnant wetlands, but they also have the lowest Shannon's diversity index. And one of the reasons that this happens is because of the site history and management practices that have been going on at these sites, and I'll explain those a little bit more in depth later in the presentation. This is the last figure. So this is a linear regression that our team ran comparing the native species richness to the topographic coefficient of variation. And basically the coefficient of variation is calculated by dividing the standard deviation of the topographic elevation points by the total number of points that we recorded at each site. So as you can see with this graph, as the topographic variation, so the more changes in elevation along the site, as that increases, we are also seeing an increase in the native species richness. So we can see that as the coefficient of variation increases, we're seeing more species inhabit these sites. So our R-squared value for this site is around 0.43, which indicates that around 43% of the variation in species richness can be attributed to the variation in topography. And in order for our results to be statistically significant, our p-value would have to be under 0.1. And ours was around 0.16. So although these results aren't statistically significant, we are seeing some really unique patterns going on between all these sites. And what's important and interesting is the fact that all of our sites are so diverse and so different. There's so many variables, but that the sheer fact that we're finding these patterns and these correlations it's really cool. It's really interesting. And then lastly, we were given a presence-absence data by one of our community partners, and what we did was find out what were the wetland indicator statuses for all the species that were located there. And what the indicator status is basically just a way to classify what types of environmental conditions plants can persist in. And these range from facultative, which is kind of an umbrella term, and these plants can persist in either an upland or a lowland conditions, those hummocks, the high grounds, and in the hollows, those low grounds. And obligates tend to be specialists, so these plants are only going to be found on either the upland or they're only going to be found on the lowland. So back to Willow Creek. This site, not only did it have the most variation in topography, but it also had the highest species richness for facultative and obligate analysis. So from this presence and absence data, we can infer that this site hosts plants that colonize the specialized niches present with high variation in topography. So, like I said, Willow Creek, kind of a strange site. It's considered high quality, yet it had a low Shannon's diversity index. Well, this site's been actively managed and restored for quite some time, and one of the methods they use to manage it is these prescribed burns on intervals from 2 to 5 years. So what this does is it kind of changes these disturbance regimens that are found naturally in these wetland sites, and it's changing the plant community composition. So what's happening is we're seeing these sites dominated by only a few native plant species, and this is having an effect on the species evenness at this site. So by having this effect, by having more cover of this tufted hairgrass, we're seeing this Shannon's diversity index being a little bit lower than some of the other sites. So now Sam's going to go ahead and take over the rest of the presentation and conclusion.
All right, I'm going to try to burn through this real quick. So bear with me. All right, so although our data were not entirely conclusive, the patterns in our analysis revealed to us consistently that they suggest in remnant wetlands, microtopography plays an important role in the presence of native plant species. Areas with a high coefficient of variation support more native species, and in addition, restored sites demonstrate a high native species richness and a low introduced species richness. We have a few recommendations, and as we are not fully conclusive in our results, we suggest more in-depth studies to connect our data to our hypothesis. That being said, sites with a high ratio of introduced species might become management priorities in the future. It's a good place to start. And in addition, we recommend the inclusion of microtopographic variation in future restoration projects. Before I wrap up, I'd like to share some of our products, because I'm extremely proud of them, that over the term helped us develop our professional skills. We created a monitoring plan which we followed in the field to gather our data. We created a website to share with the public our findings. We made a beautiful poster, which is going to be over here by our table. And we also created a final report to assist our community partners in their research endeavors. I would like to give a very warm thank you to our community partners, the Lane Council of Governments, the Institute for Applied Ecology, the City of Eugene itself, and the Environmental Leadership Program. And on a personal note, I would like to thank Jeff Kruger for showing me where to throw my resume around. Because otherwise I would be unemployed in a very undesirable location. Thank you all for sitting through our presentation and looking. And in addition, we have a potential Grammy nominee rap video, which is going to be at our table. We'll play it on our laptop for you as many times as you like, even though